So let us discuss about cultural identification. It is one of the important thing that we do in practical lab. Okay, so because we have to identify many pathogenic organisms based on various biochemical reactions. Okay, so let us discuss about cultural identification. So usually an identifier of the pathogen is done by three methods. We already know there is a routine method, identification by routine method, that is our microscopy and cultural media, and then we do biochemical reactions. Then there is identification by ID by our molecular method. In molecular method we do detect gene, okay, usually done by PCR. There are conventional PCR and real-time PCR. Then there is our identification by immunological method. Okay, in identification by immunological method, what we usually detect is our antigen or antibody, which we usually discuss in our immunology chapter. Okay, so here we discuss our routine method and identification by molecular method. So first let us discuss about routine method. In routine method, what we do here is that first we do microscopy in order to find the preliminary finding, whether it was a bacteria or a or it was a fungi, etc., or it was a protozoa, etc., or it has a different motility, different structures, chromatic granules, etc. So these all findings are done by microscopic method. After microscopy, what we do culture. Okay, we do culture it in cultural media. We do culture, then we do biochemical reactions. In this culture, we do biochemical reaction, or also we do automated system. What we do here is that after culture we do biochemical reaction in that culture, or else we detect what the pathogen under machines, that is the automated system. Automated systems are fast and very precise. Okay, these are very fast and very precise. So what are the examples of some automated system? There is Maldi Toff, then there is Whitetech 2, then there is Phoenix. Sorry, Phoenix spelling is not there. These are all, I am taking all this from Abur Shastri textbook. Okay, so the pics, if you needed, it is in the textbook. It is not detailedly needed. What we need is our biochemical reactions. Okay, so let us detailedly study about biochemical reactions. So first, when we get a culture, what we do is that we do first mainly two tests, that is the oxidase test and catalase test, because these tests are, you see, these tests can be either given by gram positive or gram negative. Both can give these tests positive. Okay, so that's why we do first one is the catalase test, then we do oxidase test, because this test can be given both by gram positive and gram negative. After these tests, what we do is, if it was gram positive, then we do test specific for gram positive. If it was gram negative, then we do test specific for gram negative, etc. Okay, so there are tests specific for, these are gram negative. Then there is also tests which are biochemical tests which are specific for our gram positive. Okay, so what are all the tests that are specific for gram negative and positive? First, let us discuss of biochemical test for gram negative. Okay, so there are mainly four tests which was for gram negative bacteria. First one is our indole test. Then there is our citrate utilization test. We will discuss detailedly about each one of them. Okay, then there is our urea hydrolysis test. Then there is our famous triple sugar iron agar test, which is an important test because we can detect many things from this test. Okay, triple sugar iron agar test. So in this module we are going to discuss about catalyst oxidase test, this all detail, okay. Then there are tests for gram positive bacteria. So what are the tests for gram given by gram positive bacteria? First one is our coagulase test. So the name itself indicates what is the coagulase test. If a bacteria contains coagulase enzyme, that culture media will going to get coagulized. That is the coagulase test, okay. For example, our Staphylococcus aureus is a coagulase positive. Staphylococcus aureus. So what happen is that in culture test tube, if you take a culture test tube of Staphylococcus aureus, there will be coagulation. Then the test is positive given by Staphylococcus aureus. If there is no coagulation, then the test is negative. This is the coagulase test. It is very simple, given mostly by gram positive bacteria. Then the next test is CAMP test, okay. It is an important test, CAMP test. Yes, no, this is not a cyclic AMP, okay. This is the name of scientist who found this enzyme. CAMP is an enzyme, like phospholipase like enzyme, okay. So what is the full form of CAMP? It is the name of scientist who found these enzymes. This is the Christie Atkin, Munch. These are the name of 4 scientists. Petersons. So CAMP test is the Christie Atkins Munch Peterson test. Though find the enzyme called CAMP. CAMP is an enzyme which is a phospholipase like enzyme. This is a phospholipase, okay. This is a phospholipase. That cause synergic hemolysis with beta hemolysin produced by Streptococcus aureus. Okay, so what is mean by that is, you see Streptococcus aureus, this is the Streptococcus aureus, okay. You see this is a streak of Streptococcus aureus. So Streptococcus aureus will produce beta hemolysin, okay. So beta hemolysin produces beta hemolysis. So when we do a streak perpendicular to the Streptococcus aureus, you see this is the perpendicular streak on Streptococcus aureus, okay, of group B Streptococcus, okay. Group B Streptococcus which produce our CAMP enzyme. Then there will be a synergic hemolysis with that of Streptococcus aureus. That is this test. There will be a synergic hemolysis along with the Streptococcus aureus streak, and this test is specified for group B Streptococcus because they produce what? They can only produce our CAMP enzyme, that is a phospholipase like enzyme that helps in the synergic hemolysis with beta hemolysin produced by Streptococcus aureus. I hope you understand what is mean by CAMP test, okay. Then our third test, it is given by group B Streptococcus. Then our third and fourth test will study together, okay. That is the heat tolerance test. Then fourth one is the bile esculin hydrolysis test. So why we study this together? Because these are both given positive by our Enterococcus. Sorry. You see, Enterococcus have high heat tolerance. So when we heat it high temperature, it will not die. That is the heat tolerance test. It is given positive by Enterococcus. Also bile esculin hydrolysis test. Because Enterococcus can hydrolyze bile esculin, so it will give an color. Because there will be an indicator for the hydrolysis of bile esculin. So there will give a color, then the test is positive. So it is given positive. These two tests are given positive by Enterococcus. Then the next two tests are also discussed together. Okay. Next two tests are inulin fermentation. and then there is bile solubility. Okay, inulin fermentation, here nothing means, here there will be inulin in the culture media, and if the sample can ferment the inulin, then there will be a color change due to indicator, that means inulin fermentation test is positive. These two tests are given positive by Streptococcus pneumoniae. Okay, Streptococcus pneumoniae is soluble in bile. So also Streptococcus aureus, you see, sorry, Streptococcus viridae, Streptococcus viridae is not bile soluble. Okay, so we can differentiate Streptococcus pneumoniae from Streptococcus viridae by bile solubility test. So these two tests are given positive by Streptococcus pneumoniae, and we can differentiate it from Streptococcus viridae by bile solubility test. Then our seventh test, which is given positive by gram positive bacteria, are our antimicrobial susceptibility test, which is usually helps to differentiate whether it is a different strain of same bacteria. Okay, so what are the example for antimicrobial susceptibility test? One example is our optokine. So optokine is an antibiotics usually for Streptococcus pneumonia. Okay, so Streptococcus pneumoniae, it is sensitive towards opkin. Then there is our Streptococcus viridae, it is resistant towards optokine. Okay, so we can differentiate it is whether Streptococcus pneumonia or it is a Streptococcus viridae, because Streptococcus viridae is resistant towards optokine. Then the next one is bacitracin susceptibility test. Okay, usually it is sensitive by group A Streptococcus. Okay, group A Streptococcus are sensitive, whereas group B Streptococcus are resistant. Okay, so we can differentiate A and B groups by bacitracin susceptibility test. So these are the main 7 tests for differentiating gram positive bacteria. So let us detailedly discuss about these 4, 5, 6 tests. Okay, so first one is our Catalase test. Catalase test can be given positive by gram positive and also by gram negative. Okay, catalase and oxidase test. So what happened in catalase test is that we detect the presence of enzyme catalase. Okay, here is the, we detect if a bacteria has a catalase enzyme, then the bacteria is catalase positive. If it doesn't have catalase, then it is catalase test negative. Okay, so usually most are catalase positive. So let us discuss about some exception of catalase negative. So what are all the microorganisms that are catalase negatives are Streptococcus, Pneumococcus, Enterococcus. These all are gram positive. Okay, these 3 are gram positive bacteria. Catalase positive can be gram positive or gram negative, but mostly catalase negative, these 3 are gram positive bacteria. So we have to study which is the less number. Okay, so most catalase are positive and most of the organism are catalase positive, and this Streptococcus pneumonia and the Enterococcus are catalase negative. So what is the mechanism here is that you see we produce H2O2 in the culture media. So what happens is that if it has catalase enzyme, it will convert into O2. So there will be bubbles forming at the surface. So it is catalase positive and this one is catalase negative. This one is catalase positive and this one is catalase negative because there is production of oxygen in here. That's why the bubble is formed. So that was all about catalase test. Then we goes into oxidase test. Okay. Second test is oxidase test. This test is also not specific for gram positive or gram negative bacteria. Okay, not specific. So what is mean by oxidase test? In oxidase test, what happens is that here we detect our cytochrome oxidase. It is an enzyme which has an oxidating capacity. Our cytochrome oxidase. Here we detect cytochrome oxidase. You see, so what happens is that in a filter paper, what we do is that we dip it in a oxidase reagent. Okay, dip it. This is the filter paper. Oxidase reagent, and we spread it to what we do is that we spread it to cultures of bacteria that we want to test. Okay, so what happens is that if a cytochrome oxidase positive bacteria will produce deep purple color here, then that is catalase then that is oxidase positive. It consists of cytochrome oxidase. If it doesn't change color, then it is oxidase negative. Okay, so this is the cytochrome oxidase test. So what we want to know here is that most of the microorganisms are oxidase negative. Most of the microorganisms are catalase positive, but most of the microorganisms are oxidase positive. For example, Enterobacteriaceae, okay, Bacteriaceae family. That isn't important. You already know most of them are oxidase negative. Then what are all the organisms which are oxidase positive? Let us study about that. Oxidase positive bacteria are example. Let us study about, okay, 1, 2. Our famous Neisseria, okay, Neisseria meningitis and Neisseria gonorrhea, meningococcus and gonococcus. Okay, this is the first set. Then our Vibrio and Pseudomonas. Okay, then there is our Bordetella and Brucella. Then there is our Campylobacter and Helicobacter. Then there is our Haemophilus. So these are the microbes which are oxidase positive, and the rest all are oxidase negative. Okay, these all organisms consist of cytochrome oxidase. So that's all about the catalase and oxidase test, which is not specific for gram negative or gram positive bacteria. Now let us go into the specific test for gram negative bacteria. Gram positive we already discussed. Then let us discuss about test for gram negative bacteria. The first test for gram negative bacteria is our indole test, right? Indole test. There will be an indicator. In indole test, what we detect is here the presence of tryptophanase. We detect enzyme called tryptophanase. Okay, we detect tryptophanase. What tryptophanase do is that, you see, the amino acid tryptophan is converted into indole by the presence of tryptophanase. Tryptophan is converted into indole, okay, by the presence of tryptophanase. So there will be an indicator, and if tryptophan is positive, then produce a reddish purple color, and if it is negative, then produce a yellow color. Okay, this is the indole test. Or positive, for some example, let us study about some example. Positive is given by Escherichia coli, then it is also given by our Proteus vulgaris. Proteus vulgaris is indole positive. Okay, then negative is shown by Klebsiella, Pseudomonas, Salmonella. Then there is our Proteus mirabilis. Okay, Proteus mirabilis is indole negative. So at least remember this: Proteus vulgaris is indole positive and Proteus mirabilis is indole negative. Okay, so this is the first test. Now let us discuss about the second test, that is citrate utilization test. It is nothing whether the bacteria can utilize citrate or not. That is the basis of this test, citrate utilization test. So if it can utilize citrate, then there will be a blue purple color. If it is cannot utilize citrate, then there will be negative color. So what are all the organism will give citrate utilization positivity? Positive is shown by Citrobacter and Enterobacter. Okay, negative is shown by Escherichia coli and Shigella. Then the third test for our gram-negative bacteria, specific for gram-negative bacteria, is urea hydrolysis test. Okay, urea hydrolysis test. So you see, urea hydrolysis test, here what we detect is whether the microorganism consists of urease enzyme. Okay, if urease enzyme is present, here we detect enzyme urease. If urease is present, what happens is that urea will convert it into ammonia. Presence of urease will convert it into ammonia. Ammonia, we already know ammonia is alkaline. Okay, so there will be an indicator, generally it is phenol red. So what happens is that phenol red in presence of what happens is that phenol red in presence of alkaline will convert it into pink in color. So if it was urease positive, then what happens there will be production of ammonia. Okay, ammonia is alkaline, so phenol red here, phenol red indicator in the medium is converted into pink in color. So if it was pink in color, then there is urease positive. If it is not pink in color, then that means urease negative. So let us discuss some examples. Okay, what are all the urease positive? Urease positive is one of the main example we already know, that is our Helicobacter pylori, because it causes peptic ulcer. It is based on this phenomena. Okay, then there is Brucella, Proteus, Klebsiella pneumonia. Okay, then which are all the organisms that give negative urea hydrolysis test? Our Escherichia coli, Shigella, Salmonella, etc. Okay, now let us discuss about our famous triple sugar ion test. In triple sugar ion test, we can detect mainly three things. Okay, whether it can produce gas, whether it can produce H2S, whether it can produce acid, that is whether it can produce fermentation. Okay, so these are all the things that we detect from triple sugar ion test. So let us detailedly discuss about triple sugar ion test. First one is that we have to know, in the name itself indicate there are three sugar, that is glucose, sucrose, and there is lactose. Okay, in the ratio of 1:10:10. Okay, Now let us discuss what we detect here in triple sugar ion test. We detect mainly three things. Okay, first one is production of gas. Production of gas. The second one is H2S production. Okay. Then the third one is our acid production. Acid production. So let us discuss. So if if a bacteria can produce gas, then what happen is here that here the medium will be lifted. Okay, medium will be lifted or it will be cracked. There will be crack in medium. Okay, our example is Escherichia coli and Klebsiella. You see, Escherichia coli and Klebsiella, when we culture in a triple sugar ion test, what happen is that there will be lifting of the media. You see, there will be lifting of the media or there will be a crack in the media due to production of gas. That is the first detection. Then if a bacteria produce H2S gas, then what happen there will be a black color. H2S gas is produced by Salmonella typhi and Proteus vulgaris. Okay, so you see this test tube and this test tube both produce H2S gas, so that's why here is a black color and also this also a black color. Now let us discuss acid production. Okay, so acid production is due to the fermentation. As we already know, the byproduct of fermentation is acid and gas. Okay, so if there is no fermentation means no acid production. The first probability is that no fermentation. Okay, no fermentation. So what happen here that you see there will be a bud and a slant. This is the bud. The basal portion is called bud and this is called slant. If there is no inoculation inside this triple sugar ion test tube, then there will be bud and slant, both color will be red. That means there is no organism. Okay. Then if there is no fermentation also, the color will be similar to that of no inoculation. That means it will be red color. Okay. The bud and slant both will be alkaline. That means no acid production, so there will be both bud and slant will be red in color. Okay, so what happen here if no fermentation occur is that both bud and slant will be alkaline, so red in color. You see, this is the one example, this one here. So the example here is Pseudomonas. Then when we talk about if a microorganism produce only glucose fermentation, if it can only happen glucose fermentation, then there will be less production of acid only. Okay, there will be only less production of acids. So what happen here is that there will be in the bud there will be production of acid, but in slant there will be alkaline. You see, this is the example. If it was only glucose production, C is the example. The bud will be acid, the slant will be alkaline. Okay, and due to acid production it will be yellow in color, this will be red in color. So if glucose fermentation, what we will get is alkaline slant and acidic bud. Okay, this will be red color, this will be yellow color. Our examples are Salmonella and Shigella. Okay. Then if the bacteria can ferment more than 2 sugars, that is glucose and lactose and sucrose. If it can ferment more than 2 sugars, then both bud and slant will be acidic. You see here, this is the one example. Both the bud and slant is acidic, that is yellow color. Example include Escherichia coli and Klebsiella. So you see, we can differentiate the organism based on three procedures here, whether it produce gas, whether it produce H2S, or whether it can ferment, that means it can produce acid. So this is the triple sugar iron test, iron agar test. So we had finished the identification by routine method, that is by biochemical reaction. Then also the pathogen can be identified by PCR, that means the molecular method, and also can be detected by immunological method, that is antigen and antibody detection. So we already told immunology we will be dealing with in the another chapter, and in molecular method let us discuss. So identification by molecular method is what will we deal with the next. Okay, so the main procedure of identification of gene causing pathogenicity is identified by molecular method. The main process is our PCR. So in PCR there are mainly three procedures. Okay, the first one is our nucleic acid extraction. We already know what you mean by nucleic acid extraction. We expose the cell into various enzymes, lytic enzymes and polyethylene glycol, etc., and protease, lipase, etc. So what happens is that we will get a nucleic acid alone. Okay, so first one is the nucleic acid extraction. After nucleic acid extraction, what we do is that we do amplification. Amplification is done in our thermocycler. Thermocycler is the instrument which we do PCR. Okay, it is done in the instrument called thermocycler. The picture is in your textbook in Apurva Shastri's. Okay, so in thermocycler we had to introduce many nucleotides, okay, primers, etc., and also Taq polymerase, etc. Okay, so you already know this mechanism. So in amplification there is three procedures. What we do, there is three procedures. First one is the denaturation, then there is primer annealing, then there is extension. So what happens in denaturation? We provide a temperature of 95 degrees Celsius. Okay, 95 degrees Celsius. So what happens here is that both the DNA strand will detach together. So there will be denaturation, and after that we provide a specific primer. That means a primer that is specified for the pathogenic gene. Okay, so what happens, this specific primer, if there is a pathogenic gene present inside the in our sample, then this primer will attach to it. Okay, that is called primer, and here the temperature is 55 degrees Celsius. And then the lastly what we do is that the extension at 72, here the 72 degrees Celsius, and it will amplify. This cycle continues on. You already know these steps. Okay, so the what does mean by if amplification is occurred, if amplification occurred, what is it means? That is, it is our predicted pathogen. Okay, how we can say that if amplification if occurred, then that is predicted pathogens? Because we are providing a specific primer. Okay, the primer that is specific to the pathogenic organism, and if only there is the presence of pathogenic gene, then only the primer will attach to the DNA. Okay, so that's why we called, if amplification will occur, then there is our predicted pathogen. Okay, if not occur, then there is not, if not occur, then that means it is not our predicted pathogen. Okay, so this is the second method, second procedure. Then after amplification we have to detect the amplification. Okay, so how we detect the amplification has occurred or not? By two methods are there. The first one is gel electrophoresis, then there another method is fluorescent. We already know this method. So in gel electrophoresis what happened? Gel electrophoresis is usually used in conventional PCR, and fluorescent method is usually used in real-time PCR. Okay, so in conventional PCR and real-time PCR there is some difference. Okay, conventional PCR we detect the quality. Okay, so whether it is present or absent, that is only we can detect from conventional PCR. We can only detect its presence. Okay, But in real time PCR we can quantify, that means how much gene, that means how much pathogen is present can be identified. Okay, we can quantify, also we can identify. Also this occurs in real time. So real time PCR are more advantage over conventional PCR. We already know RT-PCR we do in our corona test. Okay, in corona testing we actually use reverse transcriptase real time PCR. So these are the advantages of real time PCR. We can quantify, that is the main advantage. We can know how much amount of the pathogen is present in real time PCR, but in conventional PCR we cannot detect it. And also by comparing with many other methods, PCR, what we do, PCR is more sensitive and it is more specific. Why we say the PCR is more sensitive and specific? PCR is very sensitive and specific. Okay, so why we say the sensitive is because it can detect a small amount of, can amplify small amount of DNA. Okay, the PCR is sensitive in case of it can detect a small amount of DNA. It means the specific means we use specific primer here. So it will only show the if the amplification is the presence of amplification means it has the specific organism that we are predicting. Okay, so it is more sensitive and specific in this case. The next one is the identification by immunological method, which we will deal about in the chapter immunology. So till then, bye. This was a huge topic. Yeah, not that much huge. I hope you understand it. Now let us go into the next chapter.